Okay, and then so you guys all get that email with the links to the data and the collab.
So essentially, just a minutes before the start of this session, we sent out another email with not only the collab link, but also a link to data. Did everyone get that email? Okay. Good.
So once you download the Colab notebook on your laptop, you should be able to open it in Google Colab and then please make a copy. So when you click on File, there's an option that says Save a Copy and Drive. And then when you use that option, you're gonna have your own copy and you're gonna be able to like edit things in this Colab notebook, and you're gonna be able to like take notes as I go and explain like lines of code, and then also you're gonna be able to like delete lines and like try things on your own. And then so is everyone able to like download and open the link to the Colab notebook we sent out? Are we able to like make a copy of the Colab notebook? Okay, and then so Google Colab, it runs in a way so that every time it clears the data, so that in order to have access to the data, there is a link that's below the link for the Google Colab notebook in that email. And so like please download that data, and then so once you have it downloaded on your local drive, you're gonna go to this folder. So on the left side of the columns, there's one that has a little like folder icon on it. When you click on it, you can select upload data. So you can go into your downloads, and then you can select the data that we provided for you in the email. And so it's only sharing this Chrome tab, that's why it's not showing the pop-up window. But when you're uploading the data, it should be able to like prompt you to select the data that you downloaded. Is that working for everyone?
Oh, good.
If anyone has any like problems, feel free to like let us know. Oh, make sure to make a copy of the Colab notebook, or everyone will be editing this manually. Remember, it's in File and save a copy and drive, and open your own copy.
Tired though. I'm staying really late watching some
Can everyone give me like some kind of like signal if you're good with the downloading data in the Colab notebook? I'll wait for like. Okay, I'm gonna.
So
yeah.
over on that corner, is everything working well for you guys yet? Good? Oh, good? Okay, let's get started.
Okay, so I'm gonna get started. So hopefully you guys remember what Daniel covered in our first like general lecture for introduction to brain-computer interfaces. So in that lecture, we taught something a little bit about EEG, which stands for electroencephalography. And then so here we have another like more precise definition of EEG. So it is a it's an electrophysiological technique for the recording of electrical activity from the brain. And then so because it has good temporal sensitivity, we usually use it for evaluation of dynamic cerebral functioning. So that basically means analyzing more like timely brain activity instead of other modes of like BCI that have a lower temporal resolution and sensitivity. So they're less used for capturing events of your brain activity in the brain. And then so EEG devices can be built in different ways. And then so just for a brief introduction before we get into the code, there are different kinds of electrodes that have been developed. So the technology of EEG hasn't existed for very long actually. And then so the traditional or like the standard type of electrodes that we use are called wet electrodes. And they're called that because to apply these electrodes onto your head, you actually need to apply an electrolyte gel. And that's why they're called wet electrodes. And then so usually traditionally, the types of electrodes are used are silver and silver chloride electrodes. And so here you have an image of what wet electrodes may look like when we're using them for EEG. And so because it's relatively inconvenient, every time you want to use EEG, you have to like apply gel onto your scalp, and it can get into the way of like your hair and all that kind of stuff. So that's why researchers have been working on developing dry electrodes. And then so there are three major categories of dry electrodes. And the first one is gold coated single pin electrodes. I have a picture right here. And so the type of electrodes that the open BCI headset uses are actually called circular spike electrodes. So I'm not sure if you can pass it around, but I have a picture because we have it. So essentially how it looks is that you have like these spikes and they're arranged in a circular fashion. So these particular electrodes have like two concentric circles of like little spikes. And then so they contain the casing for the electrodes inside. And then the third type is solid gel based electrodes. And then actually you guys have, if you came to the EMG workshop, you will remember that we use these types of electrodes for the EMG workshop actually. That's why I've also done that here because you've seen them in the EMG workshop. And so now I'm gonna get into the specifics of the demo that we're actually working on today. So what we're gonna do today is we're gonna first describe the cognitive event that this data consists of. So what the activity that we're doing is that after we run this entire notebook, what we try to achieve is that we have this activity where a subject will select a card from 4 poker cards. And so as we described in the email, what they're gonna do is that they're going to select a certain card, keep that in their memory, and then we're going to flash one of the 4 cards randomly, 1 per second, and then we're going to repeat that 100 times. So we capture essentially 100 brain reactions to 4 categories of cards. And so with all that electrical activity recorded from the scalp, we're going to try to decipher which card they selected out of the 4. And then so that is a particular type of signal called ERP, or event-related potential, because we know those potential spikes correlate to a specific activity. And then I'll talk more about like ERP and non-ERP signals later. So now we're gonna go through exactly what we do to be able to achieve that from beginning to end. So first step here, if you have your I know it's like very small on the screen, so it's hard to read. So you better if you read like your own copy of the notebook. So the first step is we need to record EEG data with the OpenBCI headset. And then we also need to record timestamps of the card shown. And that is recorded with the same Python program that is flashing the cards. And then so actually this entire workshop is made solely of the data we collected ourselves with the OpenBCI headset. And then also all the algorithms we're developing ourselves as well. And so that is the first step where we get the data. And then so the data is, so the raw recording that OpenBCI gives us is actually a TXT file or like a text file. And so we're going to convert that to a CSV, which stands for comma separated values file. And then with that, we're going to be able to read that data again with a Python program, such as the one we're using with this Colab notebook. And then we're going to be able to interpret the data, pre-process, do ERP analysis, and then in the end, we're going to be able to make a prediction of which card do they select. First how we're going to run is downloading a necessary library. I don't know if anyone is familiar with the library MS. Very common, but it is very like widely used in EEG signal analysis and also other like time series data, such as MEG. So I'm going to run the cell and I should be able to install the library. So yeah, it says successfully installed MNE with a version at the end, so you should be able to install that. And then so the next thing that we're gonna do is we have the data and that we downloaded in the folder, and so now we're gonna read in the EEG data, and then we're gonna also read in the event timestamps because OpenBCI doesn't automatically help us record those timestamps for the 100 cards. Is everyone following? So if you have questions, please don't hesitate to ask because when I was trying to like work with the board on like executing this workshop, they also had a lot of like all kinds of questions. So feel free to ask any kind of.
Is everyone following along up to the step of installing MNE?
Do we install M&E on our terminal or do we install like within the notebook?
Within the notebook. So this cell here, when I put an exclamation mark in front, it's able to like install MNE into your present collab notebook, so that you have access within the notebook.
If you download it over terminal, I assume it's gonna download into a different environment than the collab environment that you're using. And just as a side note, we're passing around one of the spike, what is it called?
So that like spikes, they don't have a specific name, it's just like the way they look like.
Pretty spiky.
Yeah.
That's why we say it is a particularly uncomfortable piece of headset, because it's just an array of these electrodes sitting on top of the head. We have to screw them in so that it's tight. So it's pretty uncomfortable. We're good. How to install the MNE to collab.
Now I'm gonna move on, and so there is like a question that I want you guys to answer after I explain this. So we first like these 3 lines, up to here, we just import some necessary libraries also. And then so NumPy is good for processing like array data, and then you know we're using it for like analysis. And then we're also gonna import matplotlib just so that we're able to visualize our electrical activity. And then so these lines down below, you just have the file name and then the time for when this data was recorded. And then so notice this variable called labels. This labels is actually recording the timestamp structure like a list where there are 100 items in this list. And then so the first sub item, I guess inside the first every item is the milliseconds at which a card was shown. And then this number 3, so the second entry for each data point of the 100 in these in this labels variable is actually it's like a array index for the card that was shown. So in this case, I think 3, I don't remember what it was, but it corresponds to a specific poker card. So we're going to establish a data structure for the channels, and then we're going to read in the data by using like opening the file and then read in each line. And then so up to here, it's just like using for loops to repetitively read in each line and then parse it into, so each line consists of 8 variables, and then those 8 values are the electrical activity in that specific time moment for each channel. That's why we have like 8 values per line. And then so we are able to segment those and then put them into our data array. And then so we convert that to a NumPy array, so we make the later computations easier. And then here we're just checking with the printing the shape of the channels data structure and also printing the value inside the first cell. We're just checking if we read in the data correct. And so here, these 3 lines for channel names and then sampling frequency and the channel types, that's just to help M&E understand what we're doing. So here you can see that it's printing out values of what it's reading. So this parentheses 8,20899, that is telling us the structure of the data set. So we have 8 channels, and inside each of those 8 channels, we have we have 20,899 recordings. And then so that goes back to my question. What does sampling frequency mean in our case? Like, why is this significant to us? Or I guess you can think about what are the units of sampling frequency? So I can tell you it's in units of hertz, but what does that mean for our data? Anyone have a guess?
It's like essentially gonna be like how smooth you can differentiate like from point to point in time.
I think that's along the thought that I was looking for. Do you have like additional thoughts to add to that? I can tell you that hertz is in units of 1 per second. So if we say something like 250 hertz, what could that be?
Like the raw definition of sampling frequency.
250 samples per second.
Exactly. So, yeah, so what that basically means is that the EEG is able to collect 250 distinct signals for each channel per second. So actually, if you divide this number, 20,899 by 250, you're gonna get the length of the recording in seconds. So that's what sampling frequency means, and it's gonna be significant for understanding or like helping the computer understand our data. And then so now we have our data, we're gonna create an M&E object. So that's to help with the pre-processing and analysis. And then we're also gonna visualize the data. So the pre-processing steps that we used in this current workshops, we did re-referencing and a bandpass filter. So that's what you see in 1 and 2. Yeah, do you have a question?
So yeah, I just wanted to make sure, so when you say channel 0, 0, so is that measuring like the voltage level at like one specific channel, or what is that?
So when I say 0, 0, so the data is shaped like a 2D array, right? So when I say channels 0, that's essentially all the data for the first channel, right? And then so when I say 0, 0, that's the first entry of the first channel.
But what is it
So-
like physically measuring?
So I should have named the channels better, but you guys have the headset.
I passed them one of it in West Coast to check.
Oh, that's fine. I'll just show the location so that we can get a sense of like which one. Okay, so actually when you put this headset on, so this data is recorded on myself. So when you put it on, this is gonna be in the back, so you put it on like this. And then the question that you're asking 0, 0 index actually refers to channel 1, and this is actually channel 1. So the electrical activity collected from this particular electrode is what we call channel 1. And so this is channel 2. So 1 and 2 are actually looking at the prefrontal cortex. And then this has a capacity of 16 channels, so we only use 8 for simplicity. And then so I think 3 and 4 are on the top of your head, so they're like about here. And then 5, 6, and 5, 6, and 7, 8 are in the back of the head, and that is around the region of your occipital lobe. So that's where you process visual stimulus. Does that answer your question?
Yeah,
And then so before I move on to like visualizing data and like doing pre-processing, does anyone have any questions about like the previous stuff?
Is the code box running so far?
So if you're able to run this block, that means you should have the data. If it doesn't run, that means you might not have the data. And what's your question?
Yeah, so mine has an error for pulling the file. How do we,
Okay.
like, I downloaded it to the files, but not saved, so I guess.
Do you have a file over here, like in this?
Yeah, I think it might be the wrong place, I don't know.
You can click, so if you have does anyone else have that problem? Okay, so, if you click on this, like, are you guys able to like see your downloaded file in here? Oh, so you need to manually download it yourself. It's not gonna be automatically in there when you download the laptop. And once you have that downloaded, if the path is not working for you, you can, so as long as you have this file inside your folder, you can click on this right 3 dots and then select copy path. And then as you go back to where we were importing the data, you can delete this and put the path that you copied. But what I have right now should be working.
So you have it. Visual, you make it down. Okay, that works. So then, you do that. Actually,
Yeah.
You have this,
Yeah.
So the file is not in the, when you download the Google Colab notebook, the file is not going to be in there because Google Colab it sets up a temporary environment and it clears all the data every time it runs. So you're gonna have to manually download the data yourself from that email and then upload it into the Google Colab. So it's the most recent email that Ryan sent out and it's the link after the Google Colab. And once you have that in your like local drive, you should be able to upload it into Google Colab by clicking this button.
Yo.
Okay, now that, what happened that, what does it do? I had these numbers before as well, so I've been seeing this the whole time.
Just copy
I don't know how
that,
that happens, because
so just to double check. You guys should be able to see this OpenVista raw file. That should be in the link. That's in the latest email was sent to you guys. So that was just sent about like minutes ago from the workshop. So look for this CSV, look for this link to the CSV file, like it says OpenVista raw something. Download that and then upload it to this workspace for Google Colab. Just make sure it's in the root folder for this Google Colab file. So you can work with this data. That's exactly that.
Okay, is anyone still having a raw?
Working? Yeah.
I mean, we talked about this yesterday, I can't do the same thing, but like, you know,
Okay.
it's really hard, like, I forget all the restrictions and having to take like camera classes and structure as a system. They close the first one that lets you get into all the differences. So I have to, but I was which is kind of scared for some reason. I don't know, I'm just gonna go top. I want to be able to.
Guys, if the data is downloaded for you, can you raise your hand?
Like this is a race you're gonna
So whenever, okay, you can lower it. And then, okay, so if you still have issues, you can raise your hand.
Hey, did everybody hear that? She's gonna continue, but if you have problems, just raise your hand.
Actually, we should spend some time troubleshooting with everyone. It's kind of-
You have to leave in 3 minutes. I
I'm probably gonna like. It's like.
I'm gonna go ahead.
Yeah.
If you for some reason download the data into somewhere else, but it's still in collab, you can always copy your own path by like clicking on these three dots, and you can select copy path, and it should link you to wherever you download.
Oh, that's
No internet density.
All working?
Can you do it with just?
Is it working? No.
Also make sure that like, once you have the data, you want to be able to run the cells and sequences. So make sure that you install MNE first, otherwise it might give you an error for when you try to import MNE, because this is the second step.
Yeah, it's just.
Why is the first introduction?
Because electrical activity can be negative. It's a fluctuation.
Anybody else got it?
Oh, also, just like another thing, does anyone notice this number that I multiplied every entry by? Does anyone have like a guess for what that number might be doing? Like, why are we just like getting all the data that was originally in the raw file? Like, why do we need to multiply by this? Do you have a guess for why we need to multiply by this? So it's actually because of the fact that the data is not collected, the data in the raw file is not actually in terms of microvolts, which MNE is looking for. So this is correcting it to microvolts, because actually this saved data is actually a binary 24-bit integer. And then so that's why we need some processing on that. So okay, now we're gonna get into the pre-processing step. And then so the two steps that we are able to do, one of them is re-referencing and the other one is a bandpass filter. So re-referencing can be done in like different ways. So there are different like traditions for how people like to do it. And then so if you remember from the EMG workshop, we placed our ground where you have your elbow, right? So that is because that is serving as like a ground to correct for like potential, so it serves the ground, and then that is serving the same purpose in EEG. And so for re-referencing in EEG, there are different like sets of bases of references that we use. And then a very commonly used one, you might have heard of it, is mastoids. And then so those are like the bones right behind your ears. And then so because they're relatively close with other electrodes, and they should not be expected to carry any EEG data. That's why we use that as a referencing point. And then also earlobes, which is what this headset uses. So you put these two clips on your ears when you record the data. And that is the re-referencing method that OpenBCI headset uses. And then so we're gonna do an additional re-referencing where we reference every single channel to the average of all the channels. And then so that can be helpful for correcting if any channel had like mild malfunctioning during the duration of the study, so that we don't like throw out channels. Because we are only recording 8 channels right now, so every channel is like very valuable. And then so there's also a yes.
Can you kind of like explain again what re-referencing means?
Yeah, definitely. Re-referencing is basically like it's kind of like you're selecting a new ground for your signal, so that you kind of like adjust all of them by a same amount, so that you get them all onto the same scale once again.
You can probably also understand it as centering in this case.
I can show a graph actually. So
Would you say this is kind of the same as centering that you're doing?
Okay, so here I have a graph that actually shows what rereferencing does. So here you can see that, this is an EEG original reference, right? And you can see that some of them are like dipping into negatives and like positives, above 0 and below 0, right? And then so, once we do an average reference, you can see that some of them, because the average around here is gonna be something negative, so we have corrected some of them to be above 0 and some of them to be below 0. Does that explain re-referencing, like average across all channels re-referencing?
Yeah, thank you.
Yeah. And then so we also do a bandpass filter. So I want to like clarify on the idea of a bandpass filter. So I guess when I first learned about bandpass filter, I was like under the idea that we're filtering for amplitude, which is not true. So we're actually filtering for frequency. So if you like imagine a wave that's going this way, right? And then you have like a wave that's fluctuating like this. So a bandpass filter would be like crop signals that are too high and signals that are too low. It's not like we place two lines and we cut amplitude. It's actually frequency, which is your electrical activity wave is actually waves of many different frequencies layered on top of each other. So you have like low frequency waves, which have like a bigger period, and you have like high frequency waves like this. And when you layer them on top of each other, you get the electrical activity that you actually record. But then some of the very high frequency signals are actually just pure noise. So we're gonna filter that out with a bandpass filter. So if you run this cell, and here we're graphing the, we're actually graphing the, does anyone know what PSD stands for? Yeah.
Power spectral density.
Yes. Okay, very good. And then so it's showing like distribution of the signals across different frequencies, and that's what the first two graphs are showing. So first graph is showing pre-filtering, where you can see that you have like some big spikes around like 60 and then 70 and then also around 120. And then so when we do a bandpass filter from 0 to 0.5 to 30, you can see that a lot of the noise beyond the frequency that we're looking for is actually quieted. We still have like something around 60, but the issue is a lot more like reduced. And then so M&E is not like, I mean, it's a pretty good software, but like it sometimes is not the best like custom. So here is actually graphing the raw signal. So that's your 8 channels exactly the way they turn out. So the reason why some of them are cropped here is that M&E doesn't like very well adjust for the range of signals. And then so, but it still helps you like visualize what electrical activity like looks like. And then I think one of them should be pre-filtered, and then one of them is, the first one should be pre-filtering and the second one should be after filtering. And then so we did pretty much 2 of the simplest pre-processing steps here. One of them is average across all channels re-referencing, and the other one is a bandpass filter of frequency. Before I move on to give like a little FYI of other very useful pre-processing stuff, does anyone have any questions or like issues running this cell? Showing up on everyone's computer. And then so other like just like for information, there are some, we did 2 of the simplest pre-processing techniques in our cell here, but then there are also other very popular pre-processing techniques. One of them is for artifact removal. So that is because actually when you put the EEG on your head, it's gonna be, you're gonna generate spike activity whenever you're blinking actually, and then also when we have a long cognitive path, like such as when we're recording EEG when the subject is sleeping, for example, because we want to monitor for something during their sleep, then they're going to be subject to like moving their body around. Like, so there's going to be muscle movement and also eye muscle movement other than blinking. So actually those are signal generated from sources that we're not looking for because they're not the source of the cognitive event that we're actually looking for. So an ICA is called, it's an independent component analysis. And actually, you might have heard something that's very popular called PCA, that stands for principal component analysis. And then so ICA is sort of like a version of that does similar things, but it's actually quite different. So what it's able to do is that it's able to separate signal that's contributed to by different sources. So for example, there's a spike from blinking, and then there can be like spikes from muscle or sudden movement, and then there is actually the signal that we want from the cognitive task. And then so ICA is actually able to separate those signals out and then subtract them from the channels. And then so has anyone heard of PCA, like principal component analysis? And anyone like take a guess for what might be the difference between like independent component analysis and principal component analysis? Yeah, should be better questions. But like, okay, so that's all right. So PCA, it actually, what it does is that principal component analysis assumes that there is orthogonality in your data, which means that what it does is that it picks out the axes that it thinks represent the most, that create the most variance in the data. So for example, if you have like some sort of like shape here, it's going to try to find the principal axes that align those data points. But then what you have the issue you have trying to apply that to EEG data is that your data for EEG actually doesn't have that like orthogonal component. So you cannot assume that it's going to be able to be separated into the most principal components, because you might lose information when you're neglecting those later components that contribute to less of the variance. And so there's also another quite popular technique. It's called the baseline correction, and it's specifically used in like ERPs or event-related potential. So baseline correction is essentially you record kind of like a blank period of data. So before the stimulus is shown, you record a blank period of like the subject is purely doing nothing, so that you have an idea of what's going on inside their brain in terms of EEG activity when they're not subject to any cognitive task. So you subtract that from actually the stimulus that you measure. So the reason why we did not have this for this workshop is that we did like pull together this workshop pretty quickly. So the data we recorded is not perfect, but in like a professional experiment, you should be able to have a time period of EEG data where you're using it for baseline correction. Okay, so the next part is analyzing the ERP signal. Before I move on to that, does anyone have questions about the FYI? Yes.
So while they're eliminating the artifacts, how does it know what to eliminate? Do we specify what an idle link is, what a muscle twitches, or?
Yeah, that's actually a very good question. So based on what I looked up, so there is actually people who are like pay very close attention to the data quality. What they actually do is that they put like sort of like accelerometers around the eye region so that they are actually able to like specifically capture those eye blinks and the event correlated, like with the signal correlated with that. But in the case that you don't have like those things that are specifically around your eye and trying to measure the activity, the algorithm itself is going to look for those spikes that look or that represent like that look very different from the other data. So if you have like EEG waves that's like this, and then suddenly you have a spike, the algorithm is going to recognize that it's that there's a clear difference between that little time period of data and all the other like good quality data that you're getting. So the ICA works better and better when you have larger and larger amounts of data without artifacts. So without artifacts, I just mean like a time period of data without them. So it's able to like, it's kind of like self-classifying what looks weird. Yeah.
Do you have like examples of like where the ICA would be good for data versus the PCA? I feel like I don't really understand.
Let me find the picture for us. Okay. So this is actually, let me make it. I don't know if you can see, but this kind of also helps to answer like your question as well. So whenever you see this, are you able to see my mouth? Okay, so whenever you see this, like these little spikes, you know that these are not contributed to by your like cognitive activity because it looks very like distinct from your other channels, right? And then so PCA is actually not used in processing EEG signal, but I know there are examples of like PCA. It's mainly used in like processing images.
What's the classifications, so reducing dimension?
Yeah, it's for like
So-
it's for the
Sorry, so you see like in EEG data, you get time series data. So you have data that comes with a time context. It's a stream of data. And it's within the stream of data, you can look for these artifacts. So it looks like the spike that's making a blip. Whereas in PCA, however, it's like you have imagine you have a pretty high dimensional plane or like let's make it simple, like you have a two-dimensional plane. And like you have a bunch of plots, you plot a bunch of data across this plane. And then you want to make some kind of a regression over this. You want to try to find the trends about like the distribution of all of these plotted data in this plane. And then what principal component analysis is going to do is it's going to try to find the axes along which the axes in this manifold along which your data gives you the variance. Say for example, you have data that looks like this. In this case, the principal component is going to be horizontal because it's most widely distributed on the horizontal axis versus in the vertical axis, you'll just get them all clustered on the vertical. Like they're too close to each other to give you a very good variance. That's what principal component analysis does. Basically, it gives you it tries to find all of these axes where the data gives you big variances, and then gives you the top few axes along which the data gives you a lot of variance. So in later steps, you can just pick to project all of your data onto those dimensions that have to project all of the data onto those principal components, so you can reduce the dimensionality of your data for easier analysis. Because very often your data is not just going to be two-dimensional; it can be very high-dimensional. Then using by principal component analysis, you can very effectively reduce the dimensions of the data for easier analysis. I hope that's clear.
I can also try to give an example for like how I understand PCA. So if I take like a more real life example, say you have like a blackboard, right? Or if you have like this screen, and then imagine that it's in a 3D place where you can take a picture of the object of the screen from whichever like angle you want. And then you're gonna send this picture to your friend and ask them, what do you think this is? Like, what do you think this is a picture of? Right? And then naturally you would stand in front of the screen, like on this axis, and then try to take a picture of the screen here. Right? And then your friend would be like, okay, that's a screen. But what if you stand like stand over here and imagine the screen goes like this, and you take a picture from this axis or from this perspective. The picture is going to be like a line, right? The side of your screen. And if you send that to your friend, they're going to have more trouble telling what this is, right? It could be like a board. It could be like a book that you zoomed into because it's just a line, right? You don't see all the sufficient like differentiating component to tell you what this is. So it's like reducing a 3D object of a screen to 2D, and it's like the 2D version of it that gives you the most information about what the 3D object is. Right? If you take a picture from the top, you also get a line. And people also have trouble telling what that is. I hope not. But like, yeah. Does that explain what PCA is?
Yeah.
And so I'm gonna move on to ERP analysis prep. So this is not exactly like the event-related potential analysis yet, but it is like preparing for that. And there's also another pre-processing stuff that's kind of involved with this. I'll talk about it. But so now we have like this continuous streaming of EEG data, right, in all 8 channels. For like it actually goes on for like about a minute and a half. So what we're gonna do is that we're gonna segment the data. If you have taken CS classes before, you might have heard of a word called epoch. I don't know how to pronounce it, but I say epoch. But so you can actually segment the data, where what we're gonna do is that the purpose of having them repeat this activity of looking at a flashcard 100 times is so that we can take 100 segments of data. So after we have the timestamp saved, so after the timestamp of each card being shown, we're gonna segment a 1 second worth of data points. So since we have a sampling rate of 250, that converts to 250 elements from the array after that particular time. And so with that, we're gonna get 100 segments of these like 8 channels of reactions after each card. And then what we're gonna do is that we have repetition of up to 100, and the purpose is that we can take all of the segments out of the 100 that are reactions after one card, on card 1. All of the segments reacting to card 2, all the segments reacting to card 3, all the reactions to card 4, and then we're gonna average them within each category. So that tends to be the most confusing part when I try to like talk to the board about this. So you're free to ask any questions, but I try to give like a more solid example. So since they're randomly generated, I guess it might be easier if I just all use 25, but because they're randomly generated, let's say that we have 100 segments, and actually 25 of those segments of 1 second worth of data are of card 1. And we're gonna put them all of them together, and we're gonna average them. And then we're gonna do that same thing for card 2, average them, and the same thing for card 3, average them, and the same thing for card 4. And then so, yes.
How was that number chosen? Like, is it around that number where you can actually clearly differentiate, or is there like more or less?
Do you know like the number 100?
Yeah.
So that I just picked it myself, honestly. So I felt like 100 repetitions should give you like an expected value of like 25 iterations per card, right? Because you have like 5 cards. And I felt like that could have been enough, like number of samples for you to get a good average of that reaction. You could go up more, but also like for the sake of like the time of the experiment, if the number goes too high, like if you show them like 500 cards, they're gonna be tired, and their like mental reaction to the card is gonna like diminish once time goes on. So I felt like 100 was a number that worked, but also it could be up to like experiments.
And you feel like that gives good differentiation? I guess we'll see it, but is that like you can clearly see like if they're figuring out hard?
I mean, I don't have a feeling, but based on what I got with the data, I think I was able to see like a clear difference, and I can show how that works. And then so actually, after we take the average of each of these classes, that is what we call like the time during which we may observe an ERP. And then so if you run the next 2 cells, they do whatever I said in the ERP analysis prep, and then you should be able to see 8 of these graphs, and they represent 8 different channels. Are the graphs being generated for people?
Mine is not.
Yes.
Oh, okay, never mind. This is something that I didn't delete this and put it.
They're not supposed to
Everything should run until the ERP. I think it's because with the dual that we had.
Yeah, I just want to say.
Yeah, there's like some leftover code from when we were doing oh, there's a line that you need to change for a certain word.
Basically, you just highlight the line.
Oh, so you like delete what I had in this step, type this.
Yeah.
Can everyone find this line? Find this block, paint the line.
Yeah, they just search out like.
Is that clear enough?
Yeah.
Honestly, we're all right. We talked about it. I can show you that thing.
You want to see mine, eh?
Yeah, so my
Like life science and healthcare strategy, but I wanted, like, I have a lot of hard time.
I am trying to apply with science.
I need to be sure I wanted to be like. What do you do? Like, what is?
Yeah, that was. That's what the ones I were.
Oh, so would you design the programs, or you would actually be like the person who does like?
Anyway, so that's, you're definitely working.
Yeah, I was born in grade school, I was there for ground, or then relocated to the city. So it's like.
Guys, quick check. A quick check. This is the latest block working for everyone. Like up to the block where it's showing you 4 different colors curves. And then you should see 8 different blocks of that. Okay.
So I guess I can explain like the code line by line here very quickly. So we like get the data from what the M&E was filtering, and then so after all that filtering we're referencing, we get the data back from M&E again. And then so we here is when we are getting the 100 segments, and then so you can see here it says plus 250. Essentially what that does is like it cuts off 1 second worth of data, because remember we said the sampling rate is 250, right? So 250 recordings per second. And then so what we do here is that we're plotting the ERPs of the 8 channels subject to each category of cards. So what you can see here is that you should be able to see 8 plots, and on each plot you should have 4 different lines. And then if you scroll down, you should be able to see that on some of the channels you can see a pretty clear distinction in one of the signals compared to the other ones. And then so in some channels it's less obvious than others, but if you are able to like go through each of these channels, you should be able to see that. So here it's pretty clear that the blue channel, or the blue ERP corresponding to a specific card out of the 4 is looking like it's getting some pretty distinctive reactions to the cards. This channel is probably, it looks like it's subject to a lot of noise. So that's something that happens pretty frequently with like these EEG devices. So they can get like unreliable. And you can also see in this channel, so this is channel 7, and it's in the back of the head. So that is in the occipital region, where it's where I said we process visual stimulus. So it's reasonable that we expect a differentiation between this card and the other cards in this channel, because it's in the back of the head where you're visualizing the different stimulus. And then so the color coding is here. The 4 cards each have a distinct color. So in case you want. And so ERP is actually, it's not a particular type of signal. So it stands for event-related potential. And actually, there are many different kinds of ERP signal. If you actually just do a Google search for like different kinds of ERP, you're gonna get many of them. So they're essentially like a particular differentiating signal that we notice corresponding to a specific event. So the P300 is actually a pretty popular, or like a pretty famous, I guess, ERP. It's actually very widely studied for ERP analysis. And essentially what it refers to is a signal that appears about 300 milliseconds after a stimulus is shown. So that could be an auditory stimulus or a visual stimulus, or also like many different kinds of stimulus can fall into this category. But it is something that appears about 300 milliseconds after you actually get the stimulus. So that's what we're interested in right now, and that's why we're going to be interested in a segment of the, I guess a subsegment of the 1 second segment that captures that P300. So we're going to take the average of the absolute value of the signal, because you notice some of them are negative, and over the period from 200 to 400 milliseconds for each of the 4 cards. And then we're going to use so this is finally the point where we get to making the prediction. We're using something called the Pearson's correlation coefficient, and it finds it's kind of like a correlation between 2 variables. And here the 2 variables here is the time the card was shown and the P300 amplitude. And then so that coefficient is going to make our prediction, because the highest coefficient indicates that the highest P300 amplitude was observed for that particular card. So we get 4 values here, and you can see that 3 of them are around 0, whereas 1 of them is pretty distinctively positive, and that's the 3rd card, and that is the card that I chose, and I counted the number of occurrences in those 100 cards. I mean, you just have to trust me on that was my chosen card, because yeah, we can't really prove that was my chosen card, but it was what I picked. And so Oh yeah, here's another like pre-processing sort of thing that we weren't able to do in like this workshop because of the lack of time we have for preparing it. So for each of the cards, we have like many segments, right? And it is possible that for one of those segments, or like maybe a few of the segments, you have some like outlier reactions. So for example, the subject just like saw a card and missed that it was their card and remembered it later, but didn't have like a timely reaction. So you're gonna be with visualizing the EEG data for all those 100 segments and also the segments for each card. So that's 4 different categories. Usually like professionally, you would go through each of those segments and then pick out if any of them are outliers and delete them when you're considering the averaging inside each category. So yeah. Are you guys able to like run this last stuff? That makes a prediction.
You get those good values. The previous one.
Oh, and I wanted to mention that, so we are looking for some kind of distinctive signal that's occurs around 300 milliseconds after, but you can see that it's not always like perfect. And so our theoretical understanding doesn't always like perfectly translate to what we observe, because there are some like issues with the EEG stability. But I guess you can see that in this particular channel, you can see like a pretty visible dip around like 300 milliseconds, and that could potentially correspond to like what was previously studied as precisely P300. So that is the that wraps up the demo itself, and then just like some other information, because this is like a very like I guess narrow demo for what we can do with EEG. And so there are many different ERPs actually, like I mentioned, that reflect the different aspects of cognition. So we're not limited just to like visual stimulus and P300. If you look at this picture that I found from YouTube, there are actually many different kinds of signals that are correlated with a specific cognitive event. So you can do further like research on these signals yourself, but you can see that P300 here says corresponds to visual novelty. And that is what we expect with our card experiment, because when your chosen card comes up, you're getting that like visual novelty. And we can also visualize like the differentiation in our occipital lobe channels. That's 5, 6, 7, 8. And then you can also see that there are some like other different kinds of like ERPs that we might be interested in based on the specific EEG experiment that we're doing. So here are some that correspond to like attention and then alphabet recognition. That could be useful for like people who are trying to develop like EEG for like typing. And then just to give you some information for like beyond ERP. So not all signal having to do with EEG has to be correlated with a specific event, because we define event as something that's kind of like transitory. So where you're presented with something and then the event ends very quickly. There are some cases where we are interested in EEG signal and using the technology of EEG, but we're not interested in event-related potential. So some examples of that could be like long periods of data collection, where we are not looking for any particular stimulus. So for example, detecting focus. And focus is not like a spiky event that we're looking for. Instead, it's like a long period of data. And also recognizing patterns that have to do with sleep versus like a wake state. All those can be like not corresponding to a very specific event. And then also, there are also some cases, so for example, a project I've done before, we're using EEG to detect differences between subjects that one group of subjects learned their second language very early, and then the other subject group learned their second language very late. And we have them complete a writing task during the entire duration of which we record their EEG while they're completing a writing task in their second language. So you can see how that like entire period of data doesn't necessarily correspond to a specific event. And instead we're looking for like general trends and differences within those 2 subgroups. And in that experiment, we actually use like some different metrics called signal complexity. And that's something that doesn't have to do with like any specific event or stimulus. And then also EEG is also used to study the brain regions in detail. So that doesn't have to do with any particular event either. So for example, the inverse problem and also brain mapping. So these are things. So remember how we talked about EEG has good temporal resolution, right? It returns electrical activity corresponding to cognitive events very quickly, but it doesn't exactly tell you regions inside the brain that correspond to these activation of these regions that correspond to the event. So there are researchers who work on trying to enhance that resolution, and then they try to work on the inverse problem where they're trying to find out based on these surface level like scalp level electrodes, how can we find out which regions inside the brain are actually producing these signals on the scalp, so that we can have a better understanding of the brain and actually what's going on inside those regions and not just at the scalp. And also brain mapping is working towards the same effort. And also recently, they've not been focusing very much on like detecting a specific brain region that has to do with a very specific task. The shift recently has, they recently shifted focus to like developing more theories and understanding of interactions between different brain regions when a specific event occurs, or corresponding to a specific cognitive action. And then so these are like some additional resources that I consulted while like putting together, that we consulted when putting together this workshop. And so you can look a lot beyond what we were able to teach you today. So we did like a pretty simple demo of pretty much like the simplest ERP. And then so the next workshop, we're gonna look more into where Daniel is gonna teach us more about neural decoding. So once we have good quality data, what are some more complex things we can do with like machine learning models and all of that to decipher more about what's going on inside your brain. And then so you can look forward to that and also check out any of these citations if you're interested. And I'll be happy to answer any questions.
Thank you.
Thank you.
We're gonna see, I'm gonna turn this, and then just check scroll again. So if you haven't scanned it yet, just please scan it now.
Yeah, let's still take that one. I can look up